intragenic gene interaction. So interaction between the genes located on the same locus, genes located at the same locus, that is a pair of alleles, between the same genes, between the genes controlling the same character. So that's what called intragenic gene interaction. Here you get incomplete dominance, codominance, multiple alleles, pleiotropic genes. Now we are going to see about incomplete dominance, codominance only. Then other things, pleiotropic genes later. Now first about incomplete dominance. So incomplete dominance. What it is incomplete dominance? Incomplete dominance was first described by Correns. Correns who made a cross in 4 o'clock plant, commonly called Mirabilis jalapa. Mirabilis jalapa. Andimalli. Mirabilis jalapa, commonly called Andimalli. Andimandare. Andimalli. So what it is incomplete dominance when the hybrids are the intermediate between the parents due to the absence of dominance, it is called incomplete dominance. When the alleles, when the hybrids are the intermediate between the parents due to the absence of dominance. Means it is called incomplete dominance. When the hybrids are the intermediate between the parents due to the absence of dominance is called incomplete dominance. See, Carl Correns, who made a cross in Mirabilis jalapa. He made a cross between pure breeding red flowered variety of Mirabilis jalapa to pure breeding white flowered varieties. So when they made a cross between red flowered variety, when he made a cross between red flowered variety with white flowered variety, all the offsprings in the F1 generation, F1 hybrids, they are all pink flowered. And then why they are pink flowered? Actually, dominance in the sense they either red or white should have appeared in the F1 generation, but not appeared both the characters. None of the parent character appeared in the F1 generation. Why? Due to the absence of the dominance. None of the characters dominate over each other. Either red or white dominate over each other. So none of the characters dominate over each other. No characters dominate over each other. So that's why it is called incomplete dominance. Now you just see that here a cross is made between red flowered variety of Mirabilis jalapa to white flowered variety of Mirabilis jalapa. All the offsprings, hybrids in the F1 generation are pink flowered. Why pink flowered? Due to the absence of dominance. Now none of the parent character appeared in the F1 generation. So now you just see that here hybrids are the intermediate between the parents due to the absence of dominance. Hybrids are the intermediate between the parents. It's a hybrid. When the hybrids are the intermediate between the parents due to the absence of dominance. That's why it is called incomplete dominance. When the hybrids are self-pollinated, that's what when the hybrids are self-pollinated, the offspring produced in the F2 generation, the ratio of 1:2:1. 1 red, 2 pink, 1 white. Both the phenotypic ratio, genotypic ratio are the same. Phenotypic ratio, genotypic ratio are the same. Now, here, what happened here? If the genes were mixed in the sense, again, the character, which character? The white flower character, pink flower would not have appeared in the next generation. If the genes over here, only the characters are mixed, but not the genes. How can you say that? You just see that red cross between red and white, you get pink. And then again, red color, white color appeared in the F2 generation because the genes are not mixed. Only the characters are mixed. Here in incomplete dominance, only the characters are mixed, but not the genes. So this is what incomplete dominance. So incomplete, what it is incomplete dominance? Who first discovered incomplete dominance was first described by, explained by Carl Correns in 4 plant Mirabilis jalapa. Then he just made a cross between pure breeding red flowered variety of Mirabilis jalapa to white flowered variety. All the offsprings in the F1 generation, hybrids in the F1 generation are pink flowered. Why pink flowered? None of the characters dominate over each other. No characters dominate over each other. So that pink flowered appeared. It's an intermediate character. So when the hybrids are the intermediate between the parents due to the absence of dominance is called incomplete dominance. He self-pollinated the F1 hybrids and the offsprings produced in the F2 generation the ratio of 1 red to pink is to 1 white. Phenotypic ratio 1 capital R1 capital R1 2 capital R1 capital R2 1 capital R2 capital R2. So both phenotypic ratio and genotypic ratio are the same. Now what happened here in incomplete dominance? Genes are not mixed. Only the characters are mixed. That can be proved by this. Red and white cross, we get pink. Again, when the pink flower varieties were self-pollinated, the offsprings produced in the F2 generation in the ratio 1:2:1. Again, red and white appeared. So from this, it is proved that only the characters are mixed, but not the genes. Okay, it's a very important 5 mark question. I will give you notes. You just copy down the notes from that. Okay. Then molecular explanation for incomplete dominance. Normally R1 in the sense dominant. Gene, both are dominant genes given there, but they should not have used dominant genes. But in your book they are just given dominant genes. So I am using. Otherwise, only recessive gene because no characters dominate over each other. All the genes are always marked with small letter only. But here in your book, they are just given capital letter. So that's why I am giving you. But an actual thing, you must give only small letters for the genes, of course. Molecular explanation for incomplete dominance. See, functional alleles. Functional alleles R1 are functional alleles. The functional alleles, when functional alleles present in pair, so present in pair, this is homozygous condition. That functional molecular explanation for incomplete dominance. So functional alleles R1, capital R1, homozygous 1. So in this condition, what happens here? Functional enzyme is produced. This functional enzyme capable of synthesizing a red pigment. Then mutant allele R2, mutant allele, mutant genes. So the mutant genes in homozygous condition produce non-functional enzyme. So when non-functional enzyme is there, it won't produce pigment. So red pigment is not synthesized. When heterozygous or intermediate phenotype R1, R2. So only 50%, that is R1 only capable of producing functional enzyme. 50% of functional protein, functional enzyme is produced. So half of the red. So that's why you get pink color. So this was gene interaction, gene molecular explanation for incomplete dominance. Okay. Then next one about codominance. What it is codominance? See, normally codominance in the sense when both alleles express the phenotype, not alleles in the sense dominant alleles. When both alleles capable of producing the phenotype, that condition is called codominant. Codominance, both the alleles, both the dominant alleles, both the alleles in the sense, both the dominant genes or dominant alleles capable of producing the phenotype. So example for this, sickle cell hemoglobin in human, sickle cell hemoglobin in human, ABO blood group system in human, then seeds of gossypium, seeds of gossypium. So here and all you come across this. So codominant, simultaneous expression for both alleles in the heterozygote. So simultaneous expression of both alleles in heterozygote. The phenomenon in which two different alleles express the phenotype in heterozygous individual, that's what called codominance. See, blood group inheritance, ABO blood group in human. Yeah, A group, B group, ABO, AB group and O group. So these are the four types of blood group we come across this. But in there, when A group individuals married with B group individuals, you get AB together. But both are dominant one. So in that, that's what called codominance. Simultaneous expression of both alleles in the heterozygote. The phenomenon in which two different alleles express their phenotype in the heterozygous individual is known as codominance. Example: red and white flowers of camellia plant, and then inheritance of sickle cell hemoglobin, ABO blood group system in human. Here I gave you ABO blood group system in human. Say IA, A group individual. Of course, IO also there, but that's not very important. But this is only for codominance. I am giving you this. IA, then IB, B group blood. So a person with A group blood marries B group female in a sense. So in the next generation, AB blood group notice in the children. So such A group individual notice, it is due to codominance. So both alleles can express both alleles in the sense dominant alleles. When both the dominant genes express the phenotype, this is what called codominance. So now you just see that AB blood group. When such AB blood group individuals get married with AB group individuals, get married with AB group individuals in the sense, in the next generation you get blood groups in the ratio of 1:2:1. 1 A group is to 2 AB group is to 1 B group. Like 1:2:1 like that. Okay. Then next one, codominance in plants. It was described with the help of electrophoresis, chromatography. So last year we would have studied about the protein. There, the proteins are studied by chromatography. Then electrophoresis in the sense, in a vessel, one liquid medium is there, two electrodes kept in that, and then allowing current to pass through that. Actually, positive cathode, anode rods are there. Then positive charge particles move towards negative region, and then negative particles move towards positive. So in that way they move. That's what called electrophoresis. Then in electrophoresis, proteins can be isolated. Now by means of which proteins can be studied. Now for example, now Gossypium hirsutum, cotton plant. Gossypium hirsutum, Gossypium struccianum. So then Gossypium hirsutum, it has one kind of protein, one band of protein. This one other band of protein. Gossypium struccianum, another band of protein. Now when a cross is made between these two, you get both. In the hybrid, both bands, A band, B band, both the proteins are present. Both the proteins of the parents are present in that intermediate. But intermediate in the sense, but not intermediate character. Both the proteins are produced in that, but it is not intermediate. Both the proteins are present. Both the bands of protein A and B found in the hybrid. When the hybrids were self-pollinated, again the ostrich produced in the F2 generation the ratio of 1 with the A band, 2 with the AB band is to 1 with the B band, like that. Okay, this is what called codominance. The next one about lethal alleles. What are lethal alleles? See, lethal genes are lethal alleles in the sense sometimes the genes in homozygous condition cause death of an organism. It will cause, it will have killing effect on the organism. Such genes are called lethal genes. When the genes have lethality means death effect on the organism, such alleles are called lethal alleles. Such genes are called lethal genes. So lethal genes noticed in certain plants, okay, certain plants, certain animals. There are two kinds of lethal genes: dominant lethal genes, recessive lethal genes. So when the ratio of dominant lethal genes is about 2:1 final ratio, and the ratio of recessive lethal gene 1:2, like that. So that I'll just explain to you with the help of both. Okay.